Well, it's very exciting to be one of the newest members of the CABBI team and to focus in the coming years on accelerating plant gene editing, because of course, plant gene editing is such a key technology for engineering metabolism and achieving some of the CABBI goals. So I've actually been working in the field of gene editing for, believe it or not, over 20 years. In the first decade or so was really focused on developing reagents that could find specific sites in genomes, because that's really key to gene editing. You have to hone in on the specific sequence you modify. And so for many years, we focused on engineering proteins like meganucleases, zinc finger nucleases, talons. These are all classes of DNA binding proteins that we would engineer to find our target sites of interest. But the field really changed, of course, in 2012, 2013 with the advent of CRISPR-Cas systems, and they've really become the reagent of choice. And in part, and mostly because they have such an elegant targeting mechanism. They have two components, a Cas protein and a guide RNA, and the guide simply base pairs with the target in complex with the Cas protein. So CRISPR-Cas systems have been used in a wide variety of ways to modify genomes. The native CRISPR-Cas systems are basically nucleases. So after they find their target, they make a double-strand break. And we can direct the repair of that break to achieve various editing outcomes. The most common outcome is just simply letting that broken chromosome repair itself. And oftentimes it does so imprecisely, resulting in the loss of DNA at the break site. And so this is a means of targeted mutagenesis. But CRISPR systems have been modified to carry other DNA modifying enzymes. So for example, if they're fused to deaminases, they can chemically modify cytosines and adenine, and then the cell tries to repair these, and often imprecisely. So you can convert C's to T's or A's to G's. And with these base editing reagents, you can make very targeted base changes. And then a third class that's more recently been developed is the so-called prime editors, where you fuse your CRISPR or your Cas protein to a reverse transcriptase, and you augment your guide RNA so that the reverse transcriptase copies information from the RNA, copies it to DNA that then gets incorporated into the break site. And so this is a means of making a very precise targeted alteration. So achieving the full potential of gene editing in plants has been thwarted a bit by a bottleneck. And that bottleneck is the way that we deliver these reagents to plant cells. So we're using decades-old methods of using with agrobacterium or particle bombardment to deliver our reagents, often as DNA, to plant cells growing aseptically in culture. And then we identify those cells that we've edited, we allow them to proliferate, and then treat them with hormones to induce shoots and roots and ultimately the gene-edited plants. So this is time-consuming. It takes oftentimes many months. It's labor-intensive. It requires specific expertise, and many plant species and even varieties within a species do not undergo transformation and regeneration effectively. So a few years ago, my lab started focusing on a different way to deliver gene editing reagents, and that is the use of RNA viruses. So RNA viruses are widespread in plants, infecting both monocots and dicots. This is a genome of foxtail mosaic virus, and like most viruses, it's very compact and encodes all the information needed for the virus to replicate. But you can express, in this case, we're duplicating a promoter to drive expression of information that we want, genetic information, heterologous genetic information that we want the virus to carry. A drawback is this additional cargo is limited in the size. So typically you can only have less than a kb of cargo. But because CRISPR systems have two components, the Cas protein and the guide RNA, we actually create a transgenic plant that expresses our Cas protein or its derivative, and then use the virus to express that guide RNA. So one approach that we've been focusing on, again, is to take a Cas9 transgenic plant. So the plant Cas9 is expressed in all cells of the plant. Then we infect it with our RNA viral vector that expresses the guide RNA. And as the virus infects the plant, editing occurs in all of the infected cells. So the idea is we can get editing to occur in the germline, so we can harvest seeds, And then ideally a high percentage of those seeds would have targeted modifications. So we do need to use tissue culture to create the original transgenic plant, but we can reuse that plant over and over again to create a wide variety of gene edits. So we've implemented this technology in some model species, some model dicot species. This is Nicotiana benthamiana. On the left, you can see sort of a time lapse of infection. And the white sectors you see sort of in the last images, those are actually edited cells in which we've knocked out a gene in carotenoid biosynthesis. So you get this photobleaching. And this plant will continue to grow and produce, in some cases, all white inflorescences. And we can harvest seeds from these plants. And indeed, mutations are transmitted to the next generation. And in Nicotiana, over half of the seeds that we harvest from the plants have gene edits. So it's a very effective means of targeted mutagenesis. So we've got this approach to work in Nicotiana. It also works quite effectively in Arabidopsis. So my goal as part of the CABBI team is to implement this technology in the CABBI feedstocks. So we will make transgenic Cas9 expressing feedstock plants, introduce viruses. We'll switch to viruses now that infect mostly monocots, and hope to be able to collect seeds that have gene edits. So we'll focus initially on Soteria, a model plant, and then implement the technology, hopefully, if that works well, in sorghum. But of course, many of the CABBI feedstocks are more often clonally propagated. So we're going to try that tomato strategy I described, where we take and infect the plant, the Cas9 expressing plant, and then induce tillers to form through, for example, phytohormone treatment, so that hopefully we can induce shoots that could be clonally propagated that have our gene edit of interest. So as I said, it's exciting to join the CABBI team. It's a great group of investigators with a lot of expertise in plant systems biology and plant genetic manipulation. So we hope, as I said, to initially implement our technology in Soteria and sorghum, and then ultimately switch to miscanthus and sugarcane by inducing edited tillers. And then we also want to implement some of those other editing approaches I mentioned, of base and prime editing, so that through infection, we can create very targeted and precise sequence modifications. And of course, the overall goal is to really use gene editing in these approaches to accelerate the metabolic engineering efforts of the collective CABBI team. So thank you very much. I'll be happy to answer any questions that there might be in the chat.